science plays a vital role for economic development and societal well-being. Understanding this, the Academy of Sciences Malaysia was established with a mission to be Malaysia's thought leader in all areas of science, technology, and innovation. To realize its mission, ASM harnesses the nation's top scientific minds to chart the STI direction and facilitates the implementation of an innovation-led economy for the country. ASM's commitment lies firmly in fostering a culture of excellence in STI in Malaysia. ASM does this by providing independent, credible, relevant and timely STI input of national and international importance to the country. Our network of expertise comprises Malaysian scientists, engineers, and technologists who specialize in various disciplines. The Academy assists in upgrading the nation's technological capabilities in the industrial sectors by producing high-quality publications such as peer-reviewed journals, monographs, and books. In addition, ASM also provides input on current and future technology trends to be considered and taken up by the government in driving the nation's economy forward. To provide Malaysian scientists with the best opportunities and exposure, ASM actively extends its international networks and collaborations. It currently has a range of multilateral engagements with renowned scientific institutions worldwide. The Academy also champions the need to grow the right talent in STI by cultivating interests in science, technology, engineering, and mathematics to the younger generation. In short, the Academy's ethos is proudly defined as Think Science, Celebrate Technology, Inspire Innovation. science plays a vital role for economic development and societal well-being. Understanding this, the Academy of Sciences Malaysia was established with a mission to be Malaysia's thought leader in all areas of science, technology, and innovation. To realize its mission, ASM harnesses the nation's top scientific minds to chart the STI direction and facilitates the implementation of an innovation-led economy for the country. ASM's commitment lies firmly in fostering a culture of excellence in STI in Malaysia. ASM does this by providing independent, credible, relevant, and timely STI input of national and international importance to the country. Our network of expertise comprises Malaysian scientists, engineers, and technologists who specialize in various disciplines. The Academy assists in upgrading the nation's technological capabilities in the industrial sectors by producing high-quality publications such as peer-reviewed journals, monographs, and books. In addition, ASM also provides input on current and future technology trends to be considered and taken up by the government in driving the nation's economy forward. To provide Malaysian scientists with the best opportunities and exposure, ASM actively extends its international networks and collaborations. It currently has a range of multilateral engagements with renowned scientific institutions worldwide. The Academy also champions the need to grow the right talent in STI by cultivating interests in science, technology, engineering, and mathematics to the younger generation. In short, the Academy's ethos is proudly defined as Think Science, Celebrate Technology, Inspire Innovation.
Greetings from Malaysia and a warm welcome to the TropSc 2021 panel session on vector-borne and neglected diseases in the tropics. Thank you very much for joining us in this session. We have an impressive lineup of eminent panelists. For today's session, we have with us Professor Alan Coleman, Professor Dr. Sazali Abu Bakar, Emeritus Professor Dr. Jose Maria Gutierrez, and Dr. Rahman Vilayuthan to deliberate and provide us with some interesting insights on this topic. I am Yvonne Lim, a professor at the Department of Parasitology, Faculty of Medicine, University of Malaya, and I'm honored to be the moderator of this session. I'd like to thank the organizers, the Mahathir Science Award Foundation, and the Academy of Sciences Malaysia for this opportunity. There are more than 85% of the poorest poor in the world that is living in the tropics. And annually, tropical diseases, especially the vector-borne and neglected diseases, are affecting more than 1 billion people in the tropics. And these diseases are costing developing economies billions of dollars every year. Vector-borne diseases cause more than 700,000 deaths annually, and the burden of these diseases are highest in tropical and subtropical areas, whilst neglected tropical diseases affect 1 in 5 people on the planet, particularly those living in extreme poverty. Attention then must be paid to the development and implementation of new and multidisciplinary solutions and modalities for the sustainable health of the tropical populations and their environment. We are very fortunate today to have with us four world-renowned experts in key areas of vector-borne and neglected diseases. Now, in the interest of time, we will take questions only after all the four panelists have completed their presentations. So without further ado, let us move on with our first presentation. Our first panelist is Professor Dr. Alan Cowman. He is currently the Deputy Director of Science Strategy at the Walter and Eliza Hall Institute for Medical Research at Australia. He is also the recipient of the illustrious Mahathir Science Award for his excellent contribution in tropical science research. Professor Alan Cowman is the expert in the field of malaria, and his work has determined how malaria parasites evade lethal effects of important anti-malarial drugs. This knowledge has allowed detection of drug-resistant malaria parasites and to understand the prevalence and spread of drug resistance, important for developing public policy on anti-malarial use. He has developed tools to manipulate the parasite genome and constructed genetically attenuated parasites as novel avenues to develop vaccines, which has progressed to human clinical trials. And his work has identified new targets in the malaria parasite for development of antimalarials, which are currently in development. So it is with my utmost pleasure to welcome Professor Dr. Alan Cowman, who will be speaking on drug discovery and resistance, the need for novel antimalarials. Professor Cowman, the floor is yours.
Thank you very much, Professor Lim, for that generous introduction. Also, thank you for the invitation to speak to you tonight. What I'd like to tell you about is some of the work that we've been doing with respect to discovery of new antimalarials. And what I'd first like to do is give you an introduction of malaria, the problems more historically, but also the problems now in terms particularly of drug resistance. On the next slide, just back one, thanks. But, firstly, on this slide, just gives you some background on malaria. And malaria in humans is caused by two main parasites belonging to Plasmodium species, in particular Plasmodium falciparum, which accounts for approximately 150 million malaria infections per year and over 430,000 deaths annually. If not treated within the first 24 hours, it can progress to severe illness and also lead to death. Most malaria deaths are from infection with Plasmodium falciparum, and particularly occurs in children under 5 years of age. The other protozoan parasite that causes malaria is Plasmodium vivax. There are others, and Plasmodium knowlesi is one that people in Malaysia may well know. But vivax accounts for approximately 80 to 100 million malaria infections per year, and it is the most widely distributed parasite that causes malaria. And it's noted for its ability to cause recurring infection. It's proven difficult to eliminate because it has a hypnozoic form that hides in the liver. So it's essentially a quiescent form or a dormant form. And that form is particularly difficult to kill and treat using current malaria medicines. On the next slide. So there's been some really good news over the last 20 years, and particularly in the last few months with the announcement from WHO that a vaccine that's been developed against Plasmodium falciparum malaria called RTS AS01 vaccine has been recommended for deployment. So this is excellent news and will certainly save lives, particularly for children, as I said, under 5 years of age. However, unfortunately, it's not particularly efficacious, and I'll talk a little bit more about that. And clearly, we need new and better vaccines against this protozoan parasite. On the next slide. So just a bit more information on the global burden of malaria. And what you see with the maps on the right is firstly the distribution of Plasmodium falciparum, and this was essentially put together in 2017. And as you can see, falciparum malaria occurs particularly in tropical and subtropical areas. And you might ask, why are people in Australia, and the Walter Lysol Institute is right down the bottom of Australia here, why are we interested in malaria? Well, close to us and close to Australia are countries that have particular problems with malaria, such as Papua New Guinea, countries in Oceania, and also, of course, in Asia. And I've already talked about the RTSSS vaccine that WHO has announced should be deployed. But as I said, unfortunately, its efficacy is not that high. The level of protection is about 40% in children, requires 4 doses. And unfortunately, immunity tends to wane fairly rapidly. So clearly we need better vaccines if we really want to reach the goal of elimination in different countries around the world, but also eradication of malaria throughout the world. But there's some really good news about malaria. Over the last 20 years, malaria mortality rates have been reduced by 47%, and malaria incidence is down by 30%. And it's been calculated that over 4 million deaths have essentially been averted since 2000. That's the good news. Unfortunately, not so good news is that malaria appears to be resurging in some areas. And there's a few reasons for that. One is that mosquitoes have developed resistance to pyrethroid insecticides, and so insecticide-treated bed nets are not necessarily working as well as they have been. Also, the efficacy of antimalarials is not as good as it was, and particularly artemisinin-based combinations. Essentially, the parasite has been able to develop resistance, and so the use of these combinations, although still working very well in some areas, perhaps not as well as they did. So we certainly need new classes of antimalarial agents that have novel mechanisms of action, because if they have novel mechanisms of action, then any mechanism of resistance the parasite has developed will essentially not work against these new antimalarials. On the next slide. So what's made this really great progress possible? And firstly, the recognition that malaria is a global health challenge and an impediment to economic development in the countries where it is endemic in particular. And there's really been an increased political commitment and increased funding to essentially deploy the tools that we have to control and treat malaria. There are new and better tools, better medicines, and I've talked about artemisinin-based combinations, also long-lasting insecticidal nets, and rapid diagnostic tests. And as I said, substantial financial aid to essentially deploy these tools. On the next slide. However, as I said, malaria is starting to resurge, and a number of reasons, and I've mentioned a few of them, but also changing mosquito biting behaviours so that mosquitoes can shift to predominantly outdoor biting, and they can shift to biting earlier in the evening and night so that bed nets no longer necessarily protect. And that is causing an increased prevalence of malaria, but also there's increasing resource constraints due to the SARS-CoV-2 pandemic, which it is causing resurgence of malaria in some areas. There's also, as I said, drug resistance, particularly of artemisinin-based combinations, so artemisinin and its partner drugs. So we really need a robust pipeline of new antimalarials that act against novel targets to really combat this. To try and reach the goal of eliminating malaria, as I said, in different countries, but also eradication around the world. Next slide, please. So in terms of resistance to artemisinin combinations, what I've shown here in these two slides on the left is essentially the regional distribution of artemisinin resistance in 2015. And the red colour is essentially resistant parasites, and as you can see, they're mainly concentrated in greater Mekong sub-region, and starting to spread out from that, and with some occurring in Myanmar in that 2015. And we really understand, to some extent, the molecular basis of resistance to these drugs. So there are molecular markers so that you can map the geographical spread of these resistant mechanisms in the parasite population. And you see that spread occurring across Southeast Asia and to Myanmar. On the next slide, please. And what you see here is a slide of the situation in 2020 that essentially maps out drug resistance of Plasmodium falciparum to artemisinin and its partner antimalarials. And what you see in the top map is the distribution of treatment failures of artemether and lumefantrine. Artemether is a derivative of artemisinin. And so, as you can see, treatment failures have essentially spread around the world where malaria can be found. So that's a real problem. It doesn't mean that these drugs aren't useful. They are absolutely incredibly useful, but the usefulness is decreasing. And as I said, there are molecular markers to these resistant mechanisms, and you can see that in 2020, these resistant markers have really spread to the areas where malaria occurs. So prevalence of these resistant markers essentially maps to the treatment failure map as well. On the next slide. So in terms of the goal to eliminate and eradicate malaria, we really need new tools, and this is a really nice summary of review of that by Richard Feacham in The Lancet, basically outlining the reasons why we need new drugs against malaria, the sort of drugs we need, as well as new vaccines, as well as the RTSS vaccine deployment, and also new insecticides. On the next slide. So we set out essentially in collaboration with Merck Sharp and Dohme, a pharmaceutical company in the USA, to discover new drugs against malaria. Initially our aim was to understand the basic biology of the parasite and use that to identify new targets to develop these drugs, and then to develop what are called hit compounds, or identify hit compounds, develop them using medicinal chemistry to lead compounds, optimize those leads, move them to preclinical development, and then into human clinical trials. On the next slide. Medicines for Malaria Venture, or MMV, have really set a very high bar for the development of new antimalarial drugs. And essentially, this is the properties of the new drugs that they would like to be developed. In particular, CERCAT, which they require a drug that is fast-killing, long-acting, as a radical cure, and also blocks transmission of the parasite to mosquitoes. So they require, obviously, to be orally available, that it should cure as a single dose with less than 1,000 milligrams, that there be a combination of two drugs as a guard against resistance or development of resistance, that the infection be cured quite quickly, that there's a very low susceptibility to acquisition of resistance by the parasite, that it's safe, that it's simple and inexpensive, that there are no drug-drug interactions with other antimalarials or other treatments for HIV and TB, and that it's stable under proper conditions. So a very high bar. On the next slide. So as I said, we developed a collaboration with Merck Sharp and Dohme back in 2015 to use the biology that we've been analysing to develop new drugs against malaria. And so this collaborative program involved our lab in Melbourne, Australia, down the bottom, also with WuXi AppTec in Shanghai, together with Merck Sharp and Dohme in near Philadelphia in the USA. And we were able to attract funding from the Wellcome Trust in a number of tranches over the years to initially develop our biology and identify what are called hit compounds, to then develop those hit compounds to lead and optimize those leads, and then identify clinical candidates and move them to human clinical trials. And where we're at the moment is on the right here. where essentially we've identified optimized leads, clinical candidates that are moving towards human clinical trials now. On the next slide. So this is just an outline of how we do that, and it's a bit complicated, but in some ways quite simple. What we've done is essentially identify hit compounds that kill the blood stage of the Plasmodium falciparum parasite, and this is the life cycle of the blood stage, as you can see here. And then identify these hit compounds and then put them through a cycle of analysis and design so that we can design and develop new compounds that are better, more potent, that have more drug-like properties, that can be developed into what are called lead compounds. And this WM382 is one of our optimized lead compounds. Further optimized so that they're real drugs and antimalarials, and then choose a clinical candidate and move that into human clinical trials. And at the moment, we're right at the far right of this slide in terms of development of these compounds. On the next slide. And what you see here is the life cycle of the malaria parasite with the mosquito stage, and they inject what are called sporozoite forms that infect hepatocytes in the liver, and they develop there and then have a blood stage. They form sexual stages, which are taken up by the mosquito. So we needed to develop drugs that hit the liver stage, the blood stage, and this transmission stage as well in the mosquito. And the compounds that we identified with Merck, Sharp and Dohme, we basically showed that they essentially work by inhibiting two aspartic proteases, plasmepsin 9 and plasmepsin 10. And we could show that essentially those compounds that we'd identified and developed work in the liver stage to inhibit the parasite. They work in the blood stage to kill the parasite, and they work on these transmission stages to block transmission to the mosquito. So essentially we had developed new compounds that did exactly what MMV required, that they inhibit these three different stages so that we potentially could develop them further and take them into human clinical trials. So WM382, our optimized lead compound, inhibits the liver stage of the parasite, the blood stage of the parasite, and also transmission to mosquitoes. Next slide, please. Thank you. And we could also show that these compounds could be delivered orally to mouse models of malaria, and Plasmodium burgeri is one of those models, and we could show that we could cure mice with WM382 very nicely with relatively low doses of these compounds down to 3 milligrams per kilogram. And what you see here is 60 and 30 milligrams per kilogram where you get total cure. But Plasmodium burgeri does not infect humans, and so we wanted to show that it works against malaria parasites that infects humans. And so we used mouse models of humanized mouse models where we'd essentially transfused human red blood cells into the mice. And then we can infect with Plasmodium falciparum, which is the parasite that infects humans, and show that even at 3 milligrams per kilogram, WM382 cures or suppresses infection in this case. And you can show that essentially that cure or suppression of parasites is as good or perhaps even better than chloroquine, which, as many of you will know, is one of the wonder drugs of last century. On the next slide. And as I said previously, we need new drugs, new antimalarials that do not show cross-resistance with respect to other antimalarials that have already been deployed. And so what has been done here is that we have a list of malaria parasites that are multi-drug resistant to many of the drugs that are used currently, and particularly artemisinin. And what you see on the right here with green, rather than the numbers, what I've shown, green means good, there's no cross-resistance. And what you can see there is absolutely no cross-resistance with respect to WM382 killing of all of these parasites compared to these red colours, which is resistance to chloroquine, atovaquone for ATO, mefloquine, and other antimalarial drugs as well. Next slide, please. And this is essentially a figure that shows, again, the life cycle of the malaria parasite. It's a bit abstract, but I think it shows it quite nicely, where on the left here, you start with transmission to humans by the mosquito, development in the liver stage, and then the blood stage. And what it signifies is that the very large numbers of parasites where you can develop resistance is the blood stage, where you can have 10 to the 12 parasites infecting a single person. So this is where all the resistance occurs, and this is what we have to worry about. And we really need to understand whether any of our new drugs are the malaria parasite, can they develop resistance to those drugs easily? We want compounds that are very hard to develop resistance. And essentially, this spider graph on the right is a way of trying to work out whether the new compounds we developed are essentially more resistant in terms of developing resistance by the parasite, or are they easy to develop resistance by the malaria parasite? And essentially, it's looking at a number of different criteria that basically measures that ability to develop resistance. And so what we need are new antimalarials with novel modes of action, which is what we've got with these spiding protease inhibitors that have a high barrier against emergence of resistance. And these are the types of drugs that we have to develop. So the question that I want to address here is, does WM3A2 and the other clinical candidates we've developed, do they have a high barrier for the emergence of resistance? On the next slide. And essentially, this is a series of experiments that shows that the answer is that WM3A2 and the other compounds that we've developed are very hard to develop resistance to and belong essentially to a group of compounds that have been called irresistible. In other words, the malaria parasite does not appear to be able to develop resistance to it. An example of this spider graph is on the top right here, and what you see is a tovaquone, which has got a medium risk of resistance, and it's an antimalarial currently in use. And you see in blue, essentially the spider graph that outlines the resistance risk. And on the down under this is the same spider graph, but the different criteria that we've mapped out essentially by trying to develop resistance in vitro. And what you see is essentially green dots right in the middle of this spider graph. In other words, we cannot measure any development of resistance against WM3A2, which is excellent. What it suggests is that the parasite will find it extremely hard to develop resistance. On the next slide. So in terms of the compounds that we developed, where do they fit within their target candidate profiles, or TCPs, that MMV have essentially published and desire in terms of new antimalarial development? We know that the compounds we developed clear asexual blood stage parasitemia, so they're a green tick for that. We don't know if they kill hypnozoites in Plasmodium vivax. That's being tested. We do know that they have activity against hepatic schizons of the malaria parasite, so a green tick there, and they block transmission. And so in terms of their target candidate profiles, they would be excellent for treating patients, so TPP1, and also for chemoprotection, TPP2. On the next slide. So where are we at with respect to our drug discovery progression timeline? I've talked quite a bit about WM3A2, which is one of our optimized leads, but we have gone a lot further than that, and basically have compounds in preclinical development and about to enter phase 1 human clinical trials in the near future. So these compounds are very active. They have all of the desired drug-like properties, and they have the safety profiles that you need for new drugs to be used for treatment of malaria in humans. They have the pharmacokinetic properties and pharmacodynamic profiles that you really want for new antimalarial drugs. On the next slide. So what I've told you about is the problem of malaria that is still a big issue in 2021. Our work in developing new antimalarial drugs and against targets that target specifically aspartic proteases of Plasmodium falciparum, but they also work against Plasmodium vivax, and they would also work against Plasmodium knowlesi. We've shown that oral dosing of these compounds fully cure mouse models of malaria, including Plasmodium falciparum. They do not show cross-resistance to current antimalarials, and it's a very high bar for selection of drug resistance, and so we class them essentially as irresistible. They kill the 3 stages of Plasmodium life cycle that we need: the liver, the blood, and transmission to mosquitoes. And in terms of the target product profiles, they would be useful for treating patients and chemoprotection. And I haven't really talked about it, but having these compounds that we know specifically target particular aspartic proteases in the parasite means we can use them to understand the function of these proteases in the parasite. And we've been able to show that these proteases are master regulators of malaria parasite egress from the red blood cell, invasion into the red blood cell, and development within the red blood cell. On the next slide. So a program like this involves a lot of people, large teams spread within WEHI in Melbourne, which I've talked about, but also in Merck Sharp and Dome in terms of the medicinal chemistry and all of the safety and drug metabolism assays that you need to go through to develop new drugs or antimalarials against the malaria parasite, as well as our collaborators in China, WuXi AppTec, and our funding from the Wellcome Trust to develop these compounds. On the next slide. So thank you very much for listening, and I hope you found that interesting. And once again, thank you very much for the invitation to speak at this conference. Thank you, Professor Lim.
Thank you so much, Professor Cowman. I thought that was a brilliant presentation. And, the discovery of your lead compound is so encouraging. This is a very exciting prospect in terms of having a candidate that not only kills the parasite in this, the various stages in the blood, the liver, as well as to prevent the transmission. So we really look forward to this additional armory of weapons against malaria. So thank you so much again. We need to move on. So now we are moving from malaria to another critical disease that's plaguing the tropics, and this is dengue. And I'm very pleased to introduce the second panelist, Professor Dr. Sazali Abu Bakar, a fellow of the Academy of Sciences Malaysia, and he's also the director of Tropical Infectious Diseases Research and Education Center, known as TDREC in short, and the WHO Collaborating Center for Arbovirus Research and Reference for Dengue and Severe Dengue at University Malaya, Kuala Lumpur, Malaysia. He has over 25 years of research experience in emerging infectious diseases, especially in dengue and other arbovirus infections. He was the work package leader in the EU FP7 Integrated Chikungunya Research and Dengue Tools Research Consortia, and held numerous other national and international research grants. He was the editor and chairperson of the subcommittee of the drafting of the Malaysia Biosafety and Biosecurity Policy and Guidelines, and he was instrumental in establishing Malaysia's first fully certified modular biocontainment level 3 laboratory, and more recently the country's first fully certified mobile BSL-3. So let us welcome Professor Dr. Sazali Abu Bakar, and he'll be speaking to us on innovative treatments and prevention measures or methods for dengue. Professor Sazali, the floor is yours.
Thank you, Professor Lim. Thank you, the organizing committee, for inviting me to speak at this esteemed conference. I'm delighted to be associated with all these big names in the field. And allow me to have the first slide, please. I wouldn't do justice to the Ministry of Higher Education if I don't introduce you to them. Actually, the Tropical Infectious Disease Research and Education Center is one of those high COE or Higher Institution Center of Excellence under the Ministry of Higher Education. And the center was formed in 2008. And it's a center of excellence for vector and vector-borne infectious diseases and we're focusing on safety, safe science, and biosafety education, which has proved to be useful during this pandemic. Next slide, please. So as mentioned by Professor Lim earlier, for those of us who live in the tropics, in addition to malaria, dengue is another disease that has been around for a long time. For those of you who has not encountered dengue before, basically dengue is a febrile illness caused by infection with dengue virus, the virus that is transmitted to humans through bites of infected female Aedes species mosquitoes. And there are four dengue virus serotypes, basically dengue virus 1, dengue virus 2, dengue virus 3, and dengue virus 4. Next slide, please. Dengue, mostly people who are infected with dengue basically present with asymptomatic, either asymptomatic or will develop only a mild fever, and most of them will recover within a week. Less than 1% will actually develop severe form of dengue, which if it's not properly managed or properly treated, can lead to death. Yeah, death is not certain by dengue, but it can lead to death. Next slide, please. So common symptoms of dengue fever, if you look to the left there, fever, headache, muscle pain, joint pain, pain behind the eyes. These are basically the 5 most common symptoms of dengue fever. And of course, there are others who will present with rash, nausea and vomiting, and fatigue. But as you all know, that dengue fever can also develop into more severe form of dengue, which normally present with warning signs, as you can see on the right there: abdominal pain, tenderness, vomiting at least 3 times in 24 hours, bleeding from the nose or gums, vomiting blood or blood in the stool, feeling lethargic, restless, and irritable. So these are the warning signs for those who are about to develop the severe form of dengue. Next slide, please. So dengue is a global health concern now. Yeah? WHO estimates at least 96 million symptomatic dengue cases yearly, with at least another 300 million asymptomatic infections. It's difficult to calculate the actual asymptomatic infection because most people with asymptomatic infection will not go to the hospital to seek treatment. Yeah? So dengue is the most widespread mosquito-borne disease in Southeast Asia, amounting to 75% of total global dengue burden. And Malaysia alone recorded 130,101 dengue cases, with 182 deaths in 2019. But if you look to the figure on the right, you can see that Philippines, Indonesia, Vietnam, Thailand has a lot more cases of dengue in 2019. We are fortunate, I shouldn't say fortunate, but that's what it is. And with the pandemic COVID-19, it seems like the number of dengue cases have dropped tremendously in Malaysia and in the region for some reason. Next slide, please. So in Malaysia, dengue is actually not a new disease. It has been recorded as early as 1901. Yeah? And the severe dengue hemorrhagic fever and dengue shock syndrome were first recorded in 1962. Over the last decade, if you can see the bar chart on the right there, on average, every year Malaysia will have at least 80,000 dengue cases. Yeah? So the number has not subsided since 2013. Yeah? Next slide. So dengue is a major economic burden to Malaysia. Of course, not at the scale of COVID-19, but it's still a serious concern. Yeah? If you look at the data from 2020, you can see that with 90,000 cases in Malaysia and 145 deaths, with a population of only 32 million or 32.7 million, the national cost of dengue, this is conservative figure, is at 56 million US dollar. This is a very conservative figure. Some other papers have said that probably the actual number is you have to multiply that by 3. Yeah? So it's projected by 2030 with 125,000 cases. We actually have achieved that number in 2019 with 130,000 cases, but at least is with 125,000 cases predicted for 2030, the estimated deaths is at 330, and with population of 36 million, the economic national cost of dengue should be around 68 million US dollar, which is a lot of money. Huh? So as far as treatment and prevention of dengue concerned, yes, you are on the right slide just now. Currently, there is no approved vaccines for wide use and no therapeutics for specific for dengue. So before anyone jump on me, yeah, and when I say no approved vaccines for wide use, there is already a vaccine that has been approved in a number of places, yeah, but on restricted use. And I think for those who have heard about dengue, Denvaxia, there are a number of issues associated with it. So suffice for me to say at this point, yes, we do have vaccines that have gone through a proper clinical trial and have been approved in a number of countries, but still its usage is still very restricted and it's not being used in Malaysia currently. And so the only thing that we have right now is just vector control measures. And as we can guess, the vector control measure has not been very effective because the number of dengue cases keep escalating and the spread of dengue has been almost unstoppable. Yeah? So we need an alternative preventive measures that are safe, easy to use, and efficacious for dengue, yeah? Similar to malaria. Next slide, please. So studies done with Lactococcus lactis has showed that it has a potent antiviral immune stimulating effects, including against dengue virus. So studies done in vitro have shown that if we treat the cells with Lactococcus lactis, at least in cell culture, it will be able to reduce dengue virus replication. There are a number of publications that have been reported on this phenomenon. You can see one example there on your right. Yeah, and it has been replicated in vivo. And if you take mice and then you treat it with this Lactococcus, or you feed the mice with the Lactococcus lactis, and then you challenge it with dengue virus, you can see that mice that have been treated with or fed with this Lactococcus lactis are able to restrict dengue virus replication. So those were studies that were done and published much earlier, both in vitro and in vivo. Now we ask this question, could oral supplementation of diet with Lactococcus lactis prevent dengue virus infection or at least reduce dengue-associated illnesses? So next slide, please. So what we did was that we undertook a trial, a clinical trial, on Lactococcus lactis strain plasma specifically, and intake suppressed the incidence of dengue fever-like symptoms in healthy Malaysians. It is a randomized, double-blind, placebo-controlled study, and I want to take this opportunity also to declare the conflict of interest. The speaker is also the principal investigator of the trial, which was funded by Kirin Holding Limited, Japan. This trial received approval, as you can see at the bottom there, from the University Malaya Medical Center Research Ethics Committee, and also the Clinical Research Ethics Committee of Kirin Holding. And this trial was also registered in the University Hospital Medical Information Network. You see that the trial numbers are all there. So next slide, please. So basically, this trial involving two sites, involving two dengue fever hotspots in suburbans of Kuala Lumpur, Malaysia. There's two places called Desa Mentari and Mentari Kot. These are two hotspots for dengue, meaning that they have had dengue cases, prolonged dengue cases in the same place for a long period of time. Long can be up to 10 years. So these two places are well-established, well-known dengue hotspot in Malaysia. So the duration of the study was 8 weeks. And the study product, basically the active, the Lactococcus lactis, or LC plasma in short, which was formulated in tablet form and to result in 100 billion CFU per day for the volunteer to take. And a placebo is basically almost exactly the same tablet without the LC plasma content. So the primary endpoint, cumulative days positive with dengue fever-like illness symptoms. And the secondary endpoint basically to look at the average severity of dengue fever-like illness and symptoms. So this is not a clinical trial in the hospital, but it's a trial involving healthy volunteers. And it's not looking at treatment, but it's looking at basically reduction in dengue fever-like symptoms. Next slide, please. So these are the two clinical trial sites, basically Desa Mentari and Mentari Kot. So because it was a randomized study, basically we do not know the population at the beginning, but after the study we realized that the Desa Mentari Court site, the one on your left there, has a higher percentage of elderly in comparison to the Mentari Court, the study site on your right there, which has higher percentage of college students. So these are the only two differences when it comes to the study sites. If you look at the buildings, the residents, almost the same color also and almost the same height. Next slide, please. So during the time when the study was undertaken, if you look at the bar, the chart there, dengue fever during that period of time, the incidence of dengue, the green bar basically shows you the duration in which the study was undertaken, and it was at the peak of dengue season in Malaysia during that period of time. And the yellow arrow, they indicate when the imposition of the movement control order was implemented to contain the spread of pandemic 19. So the 8 weeks is just in the nick of time before the implementation of the movement control order. So that was the period in which the study was undertaken. Next slide, please. So to cut the long story short, if you look at all those bars there, those are the symptoms, the typical classical symptoms of dengue. You have headache, joint pain, pain behind the eyes, muscle pain, and fever. So what we did was that we look at the cumulative representative dengue fever-like illness symptomatic days. That means how many days the volunteers actually record that they are presenting with symptoms of headache, for example. You look at your far right there on top. So the number of days in which the volunteers reported that they have headache in the placebo group and the group, the intervention group with LC plasma, you see a significantly reduced number of days. Similarly, if you look at the bottom there, the muscle pain, those in the LC plasma group reported significantly much reduced number of days presenting with muscle pain. And then on your right, slightly to the right, in the middle there, you see that the number of days presented with joint pain was also much reduced in the LC plasma group, and also in the fever group at the bottom there, and also pain behind the eyes also a lot less in the group given the LC plasma. So the cumulative days with representative dengue fever-like symptoms were significantly reduced in the LC plasma group. Next slide, please. When you look at the cumulative symptomatic days of other clinical symptoms, not necessarily associated with dengue, you look at sore throat there on your far top end of on the right hand side. You see that the cumulative number of days of those reporting with sore throat and the LC plasma group was significantly lot less than those in the placebo group. And then at the bottom there, similarly, the number presenting with cough is also a lot less, sneezing and runny nose, vomit, and also diarrhea. So when we saw this result, we were quite happy because this result basically is in concordance to an earlier study of similar study done in Japan, in which they were looking at the impact of taking lactococcus lactis on respiratory symptoms. So we were happy that the result was reproducible in our study too. Next slide too. So when we look at the mean value of severity scores of fever for all participants, there is the chart on the right hand side there. Basically, there is a trend, but it's not significant. It's not significantly decreased. But when we do sub-analysis of the data and we look at the result as presented on the right there, result for Mentari Court. Remember, in the Mentari Court, the population has a higher number of college students, younger population. You see there was a significant decrease in the severity scores for fever for those in the intervention group. So as you see at the bottom there, in subgroup analysis, the severity of fever scores was significantly improved in the LC plasma group from the Mentari Court participants. Perhaps the younger participant of at the Mentari Court could have shown stronger immune response to the LC plasma. Next slide, please. We look at the mean value of severity scores for joint pain. Similarly, in the subgroup analysis, the severity joint pain scores was significantly improved in the LC plasma group in the mentari cord participants. So for the same reason, we think. Next slide, please. When we look at the ratio of dengue exposure and symptoms, basically the actual exposure to dengue. So if you look at the column number 2 there on your right, you see that almost no difference between the placebo group and LC plasma in terms of exposure and confirmed dengue, 5 versus 4 there. And but if you look at the symptomatic dengue presentation, the placebo group, there's 2. Of course, the LC plasma is only 1. It thinks like, wow, there is a 50% reduction, but actually the number is just too small for us to give to make any conclusion. So we need a larger study to confirm this observation. Next slide, please. So conclusion from this small study that we did is that supplementation of diet with Lactococcus lactis significantly reduced the occurrence of dengue fever-like symptoms. Lactococcus lactis or LC plasma supplementation improved the severity scores of fever and joint pain. And the good thing is that there was no adverse events were recorded on the consumption of Lactococcus lactis. Basically, this result concur with the earlier study done in Japan when they were looking at the impact of Lactococcus lactis on respiratory symptoms. Next slide, please. So for future consideration, perhaps a daily consumption of Lactococcus lactis as a supplementary alternative in dengue prevention approach during an outbreak in the dengue endemic region warrants further investigation. And as you can tell that, we need to do a much bigger study involving a lot more people for us to come to a definitive conclusion for the use of Lactococcus lactis in the prevention of dengue. Next slide, please. I would like to take this opportunity to acknowledge those who have participated in this trial, those from Tropical Infectious Disease Research and Education Center, and all of them that are listed there. And of course, Kirin Holding for providing us the fund to undertake this clinical trial and providing us with the clinical trial material, and the authorities and collaborators, the National Pharmaceutical Regulatory Agency, the NPRA, which serve the same function as FDA, EMA, and so on. So I thank them. It took us a very long time to get approval because they were not familiar with this kind of trial before. But they finally relented and approved us to undertake the study. And then Chulia Pharma for manufacturing the clinical trial product, Cellcare Clinics and Clinic Medilink for allowing us to use their facility to do recruitment of the volunteers. And of course, last but not least, the volunteers from the both sides, the Desa Mentari and Mentari Cord, for participating in this study. Thank you again to the organizing committee for giving me the opportunity to present this talk here at this esteemed conference. Back to you, Prof. Lim. Thank you.
Thank you so much, Prof. Sazali, for the interesting presentation. I think you have illustrated the benefit of using probiotics in dengue infections, and I really look forward to the further studies, perhaps in knowing even the microbiome of people that are infected versus those who are non-infected, and perhaps some novel candidates could also be known in future to be additional candidates for probiotics in terms of dengue infection. Thank you so much for that presentation. So from malaria, we've moved to dengue, and we now move to another interesting topic. Now I'll keep you in suspense while I introduce our eminent speaker. Our third panelist, who is Emeritus Professor Dr. Jose Maria Gutierrez from the Instituto Clodomiro Picado, the School of Microbiology, University of Costa Rica, a research and antivenom production center that focuses on the problem of envenomings by animal bites and stings. Now, his research interests center in the biochemistry and mechanism of action of snake venoms and toxins. He is also interested in the preclinical evaluation of the neutralizing ability of antivenoms and new inhibitors of toxins, and in the development of antivenoms for various regions of the world, as well as in the clinical aspects of envenomings and public health and social issues related to snake bites. So there you go. It's an interesting topic. So please join me in welcoming Emeritus Professor Dr. Jose Maria Gutierrez, who will be speaking on snake bite envenomings, a neglected tropical disease of high global impact. Over to you, Professor.
Very much. Greetings from Costa Rica. I would like to thank the organizers of this Congress for their kind invitation to present this topic. Next slide, please. Snakebite envenomings occur when there are encounters of humans and or domestic animals with snake in specific ecological and social contexts. In these circumstances, the snake defensively bites humans or domestic animals, causing envenomings. Next slide, please. Next slide. The global burden of snakebite envenoming has been estimated in 2.5 million cases per year, and among them, more than 100,000 cases result in fatalities, and it is estimated that about 400,000 cases result in permanent physical or psychological sequelae. And as is shown in this map, the regions most affected by this neglected tropical disease are Asia, Sub-Saharan Africa, and Latin America. Next slide, please. So snake venoms are produced in a specialized gland in the maxillary region, anatomical region of the head of the snake, and are very complex secretions which are delivered through a duct system and a fang system into the body of the victims. And there are many venomous snakes around the world, but a limited number of species are responsible for the highest number of envenomings and deaths. And in this slide, I am showing some of the most important species in the world. In the upper row, you see the saw-scale viper, the puff adder, and the najas, the cobras, which are very important in sub-Saharan Africa. And then in the lower row, you see the crate, to the left, the crate and the Russell's viper, very important in Asia. The lancehead vipers, which are the most important species in Latin America, and to the right, the taipan, which causes many cases, particularly in Papua New Guinea and other regions of Australia. So these snakes cause a very complex, owing to the complexity of the venom composition, these snakes cause a very complex pattern of pathophysiological effects. Next slide. So in some cases, like in the so-called vipers of the family Viperidae, the most important effects are local tissue damage, necrosis, bleeding, blistering, pain, swelling, as well as systemic effects related to cardiovascular alteration, hemodynamic alterations, in particular coagulopathy and systemic bleeding. Whereas in other cases, the main effect, especially in snakes of the family Elapidae, the main effects are related to neurotoxic effects with the so-called descending neuromuscular paralysis, which may end in respiratory failure. So there are different patterns of clinical manifestations of snakes. Some elapid snakes of the group of the cobras, particularly in sub-Saharan Africa, but also in Asia, also cause a severe cutaneous necrosis as it is seen in this picture. So the clinical manifestations are very different, and this brings a complexity and a challenge for the diagnosis and treatment of snake bites. The basic treatment of snakebite is the administration of animal-derived antivenoms, which are preparations of immunoglobulins able to neutralize the venom. But a challenge in the field of antivenoms is that since different venoms have different composition, antivenoms need to be specific for particular venoms in particular regions of the world. Next slide. Beyond mortality, one of the consequences of snakebite envenoming in the world, and this has been poorly documented, is are the sequelas, which are either physical or psychological. So, in the case of venoms that induce local necrosis, such as this case in South America, a person bitten by a lancehead viper, it took too long to reach the medical center and develop prominent local necrosis, which ended in amputation. At the same time, others sequela are related to neurological manifestations, chronic kidney failure, muscle problems, immobilization, and so on. In addition to that, people suffer from psychological sequela, such as post-traumatic stress disorder, anxiety, and depression. So this sequela or manifestation of snakebite, consequences of snakebite, create an expanded wave of social suffering, which affect not only the bitten person, but also their families and their communities. Next slide. It's important to remark that snakebite envenoming is also a big problem in domestic animals. And this has been recently documented by studies in Nepal and Cameroon, where a significant number of cases affecting domestic animals due to snake bites have been reported. And this have a very important impact in the household economy of these impoverished communities. Next slide. So there is a need to incorporate the One Health approach for understanding and confronting snakebite envenoming. So snake bites have to be seen within an integrated frame, and holistic frame that includes not only the effect on humans, but also the effect on domestic animals, the role of snakes in the ecosystems, and in general, the ecological and social characteristics of agroecosystems. It is within this integrated frame that we need to understand snakebite envenoming in order to develop better control systems and therapeutic approaches as well. Next slide. Snakebite as a neglected tropical disease, snakebite envenoming is clearly a disease of poverty, affecting the most vulnerable populations in Asia, Sub-Saharan Africa, and Latin America. As seen in these graphs, there is a direct relationship between snakebite mortality and low expenditure by governments in health and by low gross domestic products. So this clearly shows that snakebite envenoming and the mortality that this induces is closely related to poverty, and in particular to vulnerable populations in the rural settings, and particularly also in indigenous populations. So it has to be seen as a disease of poverty. It has to be confronted from that perspective. Next slide. Owing to the complexity of these diseases, as well as other neglected tropical diseases, of course, other aspects have to be considered for understanding and confronting this disease. For example, the phenomenon of migration, which is very important in many parts of the world. In our region of Central America, currently is a big issue. It plays a role in the incidence of snakebite envenoming because people migrating, crossing areas or regions of high incidence or high frequency of snakes usually are affected by this disease without proper attention and treatment. Likewise, it has been shown that social natural disasters such as floodings, earthquakes, and so on are closely related to an increment in the incidence of snakebite envenoming. In the same line, regions or areas suffering from armed conflicts, as in many parts of the world, are also associated with a higher incidence of snakebite envenomings. And finally, gender issues have to be closely considered when analyzing this phenomenon because a large number of bites are inflicted in women or girls, and this needs to be also properly addressed. The next slide, please. It has been discussed, and several groups have been investigating the role of global climate change in the snakes distribution and snakebite incidence. In this figure from the study of Yanes Arenas from the National Autonomous University of Mexico shows the areas of high snakebite risk in Latin America and in the Americas in general in the present time to the left, and in a prediction of the situation in the year 2050 to the right. And you see that there are regions, particularly in South America and Central America, where there will be an increase in the risk of snakebite envenoming, owing both to changes in the distribution of snakes and changes in the demography of human populations. Next slide. Next slide. So fortunately, these neglected tropical diseases that had lagged without attention for decades has now been looked upon by international health organizations and international health advocates, owing to the efforts carried out by several groups, advocacy groups, international agencies, research communities, and so on. This has impacted positively in the way the World Health Organization is uh, observing and handling this problem nowadays. So, snakebite envenoming was introduced as a neglected tropical disease in the year 2017. And in the next year, resolution was adopted by the World Health Assembly in May 2018 to raise attention and to urge governments to take specific measures and interventions to reduce the impact of this disease. And as a consequence of this resolution, the World Health Organization issued a global strategy in the year 2019 aimed at reducing mortality and sequela by snakebite envenoming by 50% in the year 2030. So this is a big step forward because this has created a global awareness of the relevance of this disease and the need to confront it with a variety of interventions in several regions of the world. Next slide. This global strategy is based on 4 pillars, and these pillars are first, to provide safe and effective treatments. And this involves not only improving the current antivenoms, but also develop newer antivenoms and newer inhibitors of these venom toxins. And also to guarantee access and availability of these treatments. The second pillar is the empowering and engaging of communities. And this pillar is based on the idea that the organization empowerment and action at the community level is very critical to confront this disease as well as other neglected tropical diseases. The third pillar is to strengthen the public health systems in countries of high incidence of snakebite envenoming. And finally, the fourth pillar is related to the establishment of partnerships, coordinations, and resources among many stakeholders in the world. So this strategy aims to reduce deaths and disability by 50% by the year 2030, and has been designed in 3 different phases: a pilot phase, a scale-up phase, and a full rollout phase. Next slide. For the implementation of this particular strategy, it is very important to promote South-South cooperation between the countries that have the highest burden of snakebite envenoming. Next slide. So I would like to remark on the need to promote this strategy within a frame of coordination and partnership, within a frame of solidarity and generosity in our global public health arena. And this partnership involves a set of stakeholders, including the research community, and the International Society on Toxinology represents this research community, including the World Health Organization and the regional offices. I myself are involved with the Pan American Health Organization in promoting several actions in this area. Also, the ministries of health in our countries, the participation of funding agencies, Wellcome Trust, for instance, has issued a big program for snakebite envenoming. The involvement of other public health advocates, foundations like the Lillian Lincoln Foundation, Kofi Annan Foundation, Hoxton Office Foundation, sectors of the media. The media is very important and has played a role in the improvement of the global awareness on this disease. The diplomatic missions in Geneva have also been very helpful for these activities. And in general, a number of organizations such as a non-governmental organization, the Global Snakebite Initiative, Health Action International, Medicine Sans Frontiers, and other organizations. So in general, the idea is to promote this global partnership under a frame of coordination and partnership and solidarity to properly confront this neglected tropical disease. Next slide. I would like again to, on my behalf and behalf of Instituto Clodomiro Picado from the University of Costa Rica, to thank you for your kind invitation and for your attention to my presentation. Thank you very much.
Thank you so much, Prof. Jose, for the excellent presentation. I think definitely the inclusion of snake bite and venoming has heightened awareness and scale up collaborative research in the areas that you have highlighted. Thank you so much. So now let's move on to our final panelist, who is Dr. Rahman Velayudhan, who is currently the head of the Veterinary Public Health, Vector Control and Environment Unit, the Department of Control of Neglected Tropical Diseases of the World Health Organization at Geneva. He is the global focal point for dengue prevention and control, integrated vector management, and he coordinates other arboviral vector-borne disease control activities at WHO. In his present assignment, he also supports the Secretariat for the Vector Control Advisory Group. He joined WHO in 1989 and has worked in Solomon Islands, Fiji, and the Philippines. And his fields of expertise include malaria control, filariasis elimination, and control of soil-transmitted helminths and schistosomiasis. So without further ado, let us welcome Dr. Rahman Velayudhan, who will be speaking on global health policy for NTD eradication. Over to you, Dr. Rahman.
Very much, Yvonne. Thank you very much for giving me this opportunity. I have actually stepped in for my director who is not available. So basically, this topic, I am really covering the new NTD roadmap, which was approved by the World Health Assembly in November last year. And this NTD roadmap sets a vision for 2021 till 2030. Next slide, please. I must thank the previous speaker, Dr. Gutierrez, for his kind words about WHO and endorsement for snakebite envenoming, which he has joined as an NTD. And as you can see, NTD consists of 20 diseases. And these diseases range from ectoparasites, protozoans, viruses, helminths. Snakebite is a new entrant, fungi, bacteria, and so on. But among all these, if you really look at it, there is, since this webinar is basically touching on vector-borne diseases, I must also highlight that 8 diseases of NTD are vector-borne. And this is where we are also looking at how we integrate work together. Next slide, please. So the new NTD roadmap is a high-level strategy, and it is basically for policy and advocacy effort. It has undergone an extensive consultation process at various levels. We can use it as a good tool. There are clear, specific, measurable targets, and it focuses on approaches to NTD across various diseases. So there are really cross-cutting elements here. Next slide, please. So NTDs also, basically we have classified these 20 NTDs into, we can classify it into 4 categories: those which can be eradicated, some of which can be eliminated, some of which may not be, can be eliminated so that they are no longer a public health problem. And then the last category is those which can only be controlled at the moment. So this is, accordingly, WHO has these definitions which are available in many books. And we really aim to eliminate NTDs as we move forward. Now, just to take a pause for a second, if we look at the last 10 years of the previous roadmap of NTD, what have we achieved? And here it is very interesting to note that 42 countries in the world has eliminated at least one NTD. 74% decline in trachoma cases. There is a 60% decline in Buruli ulcer. And 17 countries have met the criteria for filariasis elimination. So these are real achieved goals of many countries. And as we move forward to the next 10 years, we hope to accelerate this gain further. Next slide, please. So here we have very clear targets. So this is something which we really need to know, that there is overarching, where we are hoping that by the end of 2030, 90% people would require less intervention on NTDs. 100 countries would have eliminated at least one NTD, and 2 NTDs would have been eradicated from the world. And we are aiming for that. We are very close to it. 75% fewer NTD-related DALIs. Then, as I said, our new strategy aims on cross-cutting targets. There is a 75% integrated treatment or coverage of preventive chemotherapy. And we must keep in mind that over the past 3 years, the department has achieved a coverage of preventive chemotherapy exceeding 1 billion people. So 1 billion people have benefited from it, over 1 billion. Then 40% of the countries have implemented integrated skin NTDs. This is a target. Then 90% share the population at risk against catastrophic out-of-pocket payment of health expenditure. There is also 100% we hope to achieve sanitation, basic sanitation and water supply. And since this is a vector-borne, also is covered today, 75% reduction in vector-borne deaths. Now, this is a very ambitious target, I agree, because malaria contributes maximum number of deaths to vector-borne disease, but this is very well aligned with current global targets for malaria, though our current pandemic might slightly derail things a bit. Next slide, please. So we do have these NTD targets where we are hoping that the global eradication of dracunculosis or guinea worm and yaws will be achieved in the next 10 years. We also hope there will be 15 countries who would have eliminated human African trypanosomiasis, 120 countries of leprosy, and 12 from onco. Then there are eliminations. The public health problem includes Chagas, again human African trypanosomiasis. We hope to eliminate visceral leishmaniasis in 64 countries, and 58 countries would have achieved the LF targets. We want to eliminate rabies globally in 155 countries. 78 countries should have eliminated schistosomiasis. 96 countries would have less STH, and 66 countries would have eliminated trachoma. So these are some ambitious targets. And of course, we have targets for control. We really hope that one of the targets, based on the topic we discussed today, is we are trying to aim for a 0 case fatality rate for dengue. And this is achievable because many clinicians I know are of the opinion that it is achievable if we have got the trained personnel in our hospitals. And this is something we are working on. And many countries right now, the fatality is well below 1%. So dengue is on track to achieve one target, at least in terms of mortality. Then, of course, we have targets to control Buruli ulcer, the foodborne trematodes, echinococcosis, skin NTDs, and then, of course, we hope to control snake bites in 132 countries. So this is really the previous speaker has highlighted a very interesting topic on snakes. Next slide, please. So the significant change we are doing right now in the new NTD roadmap is that it is moving from accountability for impact to more measured impact. So we really want all the impact to be clearly measured. We want, instead of very programmatic vertical approach, we want to be holistic, cross-cutting approach at country level. And we also want the program ownership to be led by countries and wherever possible with the domestic financing. And this is something which we are definitely would be challenging, but we hope it is something achievable, especially in countries which have already committed to NTD elimination. Next one, please. So here we hope the three cross-cutting pillars, we have the accelerated program action, naturally the technical progress, where basically we will have better scientific understanding, more effective interventions, strategic service delivery, and we hope we will have enablers, especially for advocacy and collaboration at the grassroots level. The intensified cross-cutting approach includes integration. We are, as I said, we are integrated many NTDs under one heading called Skin NTD, and we are trying to bring more. We are also launching very soon a global arbovirus initiative to cover many arboviruses, dengue, chikungunya, Zika, and so on. Then we also want to mainstream NTDs in the national health system, bringing in more coordination with other sectors, especially the WASH sector, and in some cases even the vector control arm of the government, which may be in a different ministry. The next one is to change the operational model, where we are, as I said earlier, we want greater country ownership, clear stakeholder roles, and organizational setup to align our targets and achieve the ultimate goal of what we are aiming for 2030. Next slide, please. So here, this is a very high-packed slide. I don't expect everyone to read this, but what we have done is we have listed all the 20 NTDs on the top bar, and we have highlighted what are the challenges, where have we made technical progress, which are the ones which we know we can measure progress, the strategies and service delivery, what are the challenges, do we have problems in diagnostics, do we have problems in treatment, do we have problems in monitoring and evaluation. So all these are coded in such a way that we know which one to address them. So these are available in the NTD roadmap, so please read them. I don't want to go through them. They are quite small, and they are very elaborate to really highlight the real gaps in the current NTD portfolio. Next slide, please. So moving forward, we have identified that there are common gaps across all the NTDs, one of which is effective diagnosis. There is also a lack of medicines and effective supply chain management. There is a need for robust monitoring and evaluation system, and at a country level, it is probably better to integrate it with the health information system. And of course, NTD, neglected as is, we need more advocacy and resources of funding. Next one, please. So what we have done also is that in the cross-cutting approaches, we have really have a strategic input where we need to put more emphasis on. We have created activities such as planning, advocacy, capacity building, social mobilization, and so on. Intersectoral coordination is clear, and we are working very closely with other NGOs. We are bringing the WASH sector in, and they are very actively involved in our activities. Vector control plays a significant role, and we have the global vector control response adopted by the World Health Assembly in 2017 after Zika, which really lays the ground map for how we can integrate and work together. Then, of course, we are addressing the education sector and other sectors wherever possible. So this is what this slide is all about. Next slide, please. So basically, the next slide is the framework for the NTD. We hope we will have the political support. We will be closely monitoring the epidemiology of the disease and also looking at the social sector. The non-health sector would be involved. We will definitely focus on strengthening health systems, and especially with the post-COVID era, there is a lot of opportunities of cross-cutting interventions aligning with universal health coverage wherever in most of the country plans. So this is where we are moving forward to. Next slide, please. So we had done this basically from a national perspective. You can use this NTD roadmap to prepare and organize draft targets and strategies. You can really consult your key stakeholders and partners, and today we have to redefine some of their roles because of the limitation in resources, and post-COVID era would be a bit challenging, and we may have to refine plans and action needed as we move forward. Next slide, please. And in all these, we have really kept the people and community at the center. We are working with a huge number of stakeholders, As you know, many of you may be involved in WHO meetings, because WHO relies a lot on experts like you, and expert advisory group meetings. Many of you are invited. We make sure there is a complete balance geographically, and also from gender point of view in every meeting. And what we are trying to solicit here is the best technical advice needed for the country, especially for all these diseases. So at the end of the day, the country has to take the ownership, and that is why we are highly people and community oriented. Next one, please. So this is more application of this framework. We can put a sustainable working group, conduct stakeholder meetings, assess the sustainability of the program over a long period of time. We need to develop a sustainability plan and make sure that this is implemented and monitored over a given period of time. Next one, please. So we really have this NTD roadmap is based on several indicators. There are 36 core indicators, 34 additional disease-specific indicators. There is qualitative monitoring also, and gap analysis. We are also bringing in gender and equity into this. And so there is a complete section on monitoring and evaluation within this framework. Next one, please. So here are some companion documents. So along with this NTD roadmap, we have developed a companion document, which is the sustainability framework. An investment case is being developed. There is a monitoring and evaluation framework already. We have a second edition of WASH and NTD strategies. There is an NTD research blueprint, which is currently in progress. We are also having a companion document on NTD and One Health under development. And of course, the global vector control response. We are also bringing a small document on how to align this across NTD. I think that's my last slide. Move on to the next one. Thank you very much.
So much, Dr. Rahman, Yudhin. I think that was a brilliant presentation, and it's so apt that we are ending our session with your talk on global health policy for NTD eradication. I'm very glad that in many sense, I think the various presentations highlighted the many positive progress that we are collectively making in the areas of vector-borne and neglected disease, tropical diseases. So, ladies and gentlemen, with the conclusion of the 4 presentations, we will now start the question and answer session. And thank you for submitting some of the questions. So do continue to post the questions in the Q&A section while I start off the discussion. So if I may, Dr. Rahman, I would like to start with you first. Now, you mentioned about the 3 shifts: measuring impact, holistic cross-cutting approaches, and country ownership and domestic financing. This will require quite a shift in sort of mindset at individual, organizational, state, and country level to take ownership of this challenge, this issue. So on one hand, I think it's so important to have ownership over this, encouraging domestic financing versus the bilateral funding, and also incorporating the entities in government policies. But perhaps there may be a high chance of the current pandemic, like you said, derailing this effort due to the economic constraints faced by many countries where entities are most prevalent. So perhaps you can share with us, how do we mitigate this, as sufficient financing resources is so paramount in our efforts to eradicate NTDs? If I may have your thoughts on this, Dr. Rahman.
Thank you. Yeah. So basically, we need to understand that from the World Health Assembly, which was held actually in November last year, there was a resolution supporting the NTD roadmap. And what really this shows is that countries are committed. So basically, we had over 70 countries actively participating in the deliberation. They are committed even during the COVID situation, because NTD is really not a very expensive exercise. Eliminating NTD is feasible. A significant amount of NTD elimination, the diseases are based on drug therapy or preventive chemotherapy, which is donation drugs, which are given by companies, and we have commitments till as long as the medicines are needed to eliminate the disease. So that is quite encouraging. Now, it is only the operational funds at the country level where we need a little bit of buy-in, and there are countries who have done that. There are even countries who are buying their own drugs as and when needed. So that level of confidence the countries have got already. We do hope there will be some bilateral support, because there are bilateral agencies who have offered to continue the support, though the level of support may be varying. So I agree. There is a challenge in the next few years, but at the same time, the countries are committed, and we are also have a mandate to report to the World Health Assembly once in 2 years on the progress made. So that will be the time where more deliberation, more commitments are possible. So these are the ways in which we hope to overcome the challenges as we move forward. Over to you.
Thank you so much for giving us so much hope in that. The next question, perhaps I will open up to both Dr. Rahman and also Prof. Jose. I think you'll talk a lot about collaborations, working together, that partnership. So in your view, do you think a strong alliances of countries in the tropics? And Prof. Jose, I remember you talk about the South-South collaboration. And this is where the burden of NTDs is most felt. And how do we utilize this platform effectively and in the most impactful manner? Could I have your thoughts on this? That sort of how we heighten that collaboration. Yeah.
Sure. I think this is a very important point, as I remarked in my presentation, the South-South collaboration. And this can be developed in many different ways. And the research area, it can be also promoted in the generation of new drugs and improving of antivenoms in our field, in community-based studies, and in other areas as well. I will give you an example in our region in Latin America. Since the last decade, we have established a network of public antivenom manufacturing laboratories and quality control laboratories, which includes laboratories and groups from Mexico, Costa Rica, Panama, Colombia, Venezuela, Ecuador, Bolivia, Peru, Argentina, and Brazil. And this network has been working and is now part of a RELAPA, which is a grouping of antivenom manufacturing laboratories within a frame of solidarity, generosity, openness. So, for instance, we share technologies, we organize workshops where we share our developments, our problems. We do collaborative research projects. We promote actions in our ministries of health. Technicians and professionals from our groups visit other groups. So this is a clear example that partnership can be developed, and it has a very powerful effect it has had a very powerful effect on the improvement of antivenom quality, quantity, and also availability and accessibility in our region. So I do believe that these sort of partnerships should be organized in different regions of the world. As another example, we have established a close relationship with the Federal Ministry of Health of Nigeria. And in our laboratory in Costa Rica, we manufacture an antivenom which is being used in several countries in sub-Saharan Africa. And we are promoting these partnerships with sub-Saharan African countries as well. So there are examples of these partnerships, and I'm pretty sure in the field of neglected tropical diseases, there are many more examples. And I do believe that this should be promoted on a global basis.
Thanks so much. That's excellent. Dr. Rahman, if you would like to add, please feel free.
Thank you. I just wanted to add with what Professor Jose has just said. Basically, this, again, snakebite area itself, the partnership is fast growing, and WHO has been developing a global atlas of snake distribution and maps of incidents, and also the places where antivenoms would be available and things like that. These are work in progress. And I'm sure Professor Gutierrez knows a lot of our work, which is ongoing in parallel. And we are also lucky to get some support from the Wellcome Trust to initiate some of these work. We are also developing TPP for snake venoms, snake antivenoms. So basically, these activities are currently in progress, and a lot of work is being done. all with partnership with several research institutions. There are two other ways in which we can foster partnership, I would say, especially on the research side. One, especially since we are talking from Malaysia point of view, ASEAN is an excellent umbrella where you can foster partnership, and you have set examples for that. So you, within the 11 member state countries, you can develop very good linkages with research laboratories across the countries. That is one. We have basically that is being more or less duplicated in Southeast Asia, where the Southeast Asian countries requested WHO to coordinate the research partnership. And we had 2 meetings on that, basically to coordinate what are the top priority research and who does what. And these research institutions then collaborate with each other. There are twining arrangements. And so these are activities in progress in whole parts of Asia. Similarly, we have few activities in Africa going on, coordinated by our regional office in Africa. So there is a whole range of partnership. Apart from that, stepping below, we have several NGOs, our non-governmental organization networks, with whom we are working very closely on NTDs, and they will be playing an active role. Actually, you know that on January 30th we are going to mark the first NTD day, because it has been endorsed to have a global World NTD Day from next year by the World Health Assembly. So then the coming year we will have the first celebration, and you will see the whole array of partners coming together, and this will grow over the years to come. Thank you
so much. We are still at the topic of regional level, but perhaps this next question is for Prof. Alan Coleman. We actually have a question from the participant. So malaria parasites recognize no boundaries between countries. So what should be done by the government at the regional level to minimize the drug or vaccine resistance of malaria parasites? I think your candidate is an excellent candidate for resistance. Perhaps what can be done at regional level?
Yeah, thank you for that question. I think drug resistance in malaria will always be an issue, because the parasite has this amazing ability to develop resistance to just about anything that you throw at it. I think the important aspect will be that we always use combinations now. And at the moment, it's generally a combination of 2 drugs, but in future, it may well be a combination of 3 drugs that will hopefully prevent development of resistance that seems to occur fairly rapidly, unfortunately, with new anti-malarial drugs. So in terms of trying to prevent resistance regionally, I think it's important to tap into the new pipeline of drugs that are coming along, and hopefully use ones that have a new mode of action, and therefore there is no resistance already developed in the field, and combinations of at least 2 and perhaps 3 drugs that perhaps prevent the production of resistance by the parasite. Thank you.
Thank you so much, Prof. Coleman. Another question. So recently you're speaking about the WHO endorsed vaccine, the RTS-AS01. We know that it's not foolproof. It's not fully effective. And how do you envisage that your candidate, the WM382, if found to be very successful in the clinical trials, how would these be working in tandem with the availability of the vaccine? As what we can see now with the COVID pandemic as well, many people are vaccinated, but yet we need to have the drugs. So how would that interplay be envisioned? Yeah.
I think the RTSS vaccine, which hopefully will be deployed soon, will certainly save a lot of lives. But as we know, it's not that efficacious, and we certainly need improvements and probably combinations with blood stage antigens, along with the major component of the RTSS vaccine, which is circumsporocyte protein. I think that's really key in terms of moving towards elimination and eradication of malaria, I think drugs will, anti-malarial drugs will play a key role in that as well. And the new drugs that are being developed, which are much more irresistible, if that's what you would like to call them, will be extremely important as well. And they'll need to be deployed in a way that hopefully can mop up malaria that's arising around the vaccine deployment at the same time. So I think compounds like WM382 will be really important in terms of eliminating malaria in different regions.
Thank you so much. I think in the interest of time, we have another question that we can take, and perhaps this is from Professor Zali. Now, many countries, as you mentioned, we have seen the low rates of dengue since the start of the pandemic. What are your postulations on the science behind this phenomenon? Do you think that's a sort of whether this is an expected waxing and waning of the dengue pandemic patterns, or there is more to it? Yeah.
That's a tough one, Professor Lim. Well, we published earlier that in our studies, we identified that one of the key important reasons or factors that may affect the dengue is mobility of populations. So that's when we published the work, it was before COVID-19. But it seems like during COVID-19, especially in Malaysia, when we introduced the movement control order, and in which we restrict mobility of population at the strictest level, and the number of dengue cases just dropped. So it shows to basically is in agreement of our earlier study. It shows that the importance of mobility of population is spread of dengue.
Thank you so much. I think it makes a lot of sense, yeah, when people are not moving too much, and you know, the host is not available in terms of transmitting the infection. Thank you so much for bringing that up. Now, before I end, I'll just like to just go one round. If you have any last words, yeah, perhaps Prof. Alan Coleman, if you have any last words.
No, not really. Just thank you very much for the opportunity to speak at this conference. I think it's been great to speak in a symposium that is very broad. So I've actually been able to learn quite a bit myself about some other aspects of NTDs, which has been fantastic. So thank you very much.
Thank you so much. And Professor Zali, any last words from you?
I think in our effort to control or to eradicate or eliminate NTDs, sometimes I think we have to start thinking outside the box and looking at the fringe of our normal understanding of the disease to look at solutions. If you look at the goal of WHO to reduce the CFR, case fatality rate, to zero, I think we just need a slight notch to actually achieve that, and it's achievable within the time period that has been specified by WHO. Yeah.
Thank you so much for that. Emeritus Professor Dr. Jose Gutierrez.
Well, I'd like to again thank you very much for the invitation, and I would like to remark on this topic of alliances and partnerships, and the need to move at different levels from global organizations at the WHO to regional WHO organizations, in our case, the Pan American Health Organization. A very important role for the local governments and in strengthening of the public health systems to confront neglected tropical diseases. An important role for the research community and strengthening research and technological development, particularly in the global south. And no less important, the role of the community organization and the civil society organization for confronting these diseases. So we need a strong global partnership of many different stakeholders to understand and to confront these very complex diseases and challenges.
Thank you so much. And Dr. Rahman, any last words from you?
Thank you. I think the key to this is, first of all, thank you for inviting us. This is a great opportunity. And the key still remains that we are a neglected group of diseases, and what we can do if we want to remove the neglect, we really need to work together. Just like what Professor Gutierrez said, we need to work together in partnership at all levels possible, use the resources, and make sure that we are one entity working together. And once that movement begins, we also need to leverage on post-COVID facilities which may come along the way, the lessons learned as we move forward, embark on innovations, and see how we can all integrate our effort. And this is something which I think would come in the near future. Thank you.
Thank you so much. So with that, ladies and gentlemen, we have been very privileged today to be part of this rich discussion on vector-borne and neglected diseases in the tropics, and covering the aspects of eradication of vector-borne and neglected diseases through improved scientific understanding, utilization of cross-cutting approaches to implement interventions, heightening accessibility and equitable allocation and distribution of treatment and integral interventions. So a heartfelt thanks to all our 4 distinguished panelists for such an engaging and insightful discussion, and for their support and graciously accepting our invitation to be part of today's panel session. Our deep appreciation to all you participants and audience for taking the time to join us in today's session. Last but not least, a very big thank you to the working committee who has been working tirelessly behind the scenes for making this session a great success. We all hope that you have enjoyed the session, and please stay tuned for the next event on the presentation and adoption of Kuala Lumpur Communique on the Tropics, which is scheduled to happen in a few minutes' time, I believe in just 2 minutes' time, at 9:45 p.m. Malaysian time. So thank you very much, and stay safe, everyone. Thank you.
Thank you.